In this lesson, we'll do a brief sort of a history of life on Earth, highlighting the two endosymbiosis events and their significance. Well, it all begins a long time ago, some 4 billion years ago, with chemical evolution. Now, that process, chemical evolution, refers to how chemistry at some region on the Earth was producing really important organic molecules, the molecules that make up cells, and that cells assembled from these chemicals that were being produced. So chemical evolution, possibly in small chambers within hydrothermal vents, produces a membrane-bound metabolic engine with the capacity for reproduction using DNA as a hereditary molecule. Now that's a mouthful, but it just means the first cells emerged from chemistry. So we call this chemical evolution produced the first living things, cells. Now these are bacteria type cells, and they go on to evolve into lots of different kinds of bacteria because cells can reproduce, but when they reproduce, sometimes there are mutations, changes to the DNA, which means the recipes for proteins are being changed, so new proteins are being produced over time, and cells will have new characteristics. So we get the evolution of many different kinds of bacteria on the early Earth. Now here comes the first really important endosymbiosis event. A large bacteria incorporates a smaller bacteria, the ancestor of mitochondria, breaking the energy barrier, the evolution of larger size and greater complexity now possible. Very important process here. Were it not for this endosymbiosis event, there would be no large complex living things on Earth. It'd be all bacteria types of cells. So with the incorporation of this cell into the larger cell, and the cell continues to produce energy and it can reproduce, now this cell has the potential to grow larger in size and get more complex, and in fact evolve into cells that have more active lifestyles, like hunting cells. What's the evidence for this endosymbiosis event? Here we have the mitochondria, the size of bacteria, DNA like bacteria, reproduce like bacteria. Now, once a more energetic and larger and energetic cell evolved, it turns out there were several other endosymbiosis events, but here we'll focus on this one here, which involved the incorporation of a photosynthetic bacteria, like today's cyanobacteria. That bacteria got incorporated, it did not get digested, and this would be the ancestor of chloroplasts. So we have the origin of mitochondria, the origin of chloroplasts, and then some of these cells are going to go on to evolve into multicelled organisms like plants, and some will stay single-celled. Again, what's the evidence for this endosymbiosis event? Chloroplasts are the size of bacteria, DNA like bacteria, reproduce like bacteria. Now, the energetic cells here, the larger energetic cells, were able to then form multicelled organisms. And this happened many times actually in the history of life on Earth, suggesting it was fairly easy to do. Some mutations, some changes allowed cells to stick together, forming a larger colony, and presumably that had advantages to survival. If you're bigger, you can't be eaten by a single-celled predator, right? So now we have the origin of multicelled organisms, but again, they would be impossible were it not for that first endosymbiosis event that allowed for the breaking of the energy barrier. Now cells could get big and complex, and that made multicelled life possible in the future. So together, it's an incredible story. In the 4 billion years of the history of life on Earth, starting from simple bacteria cells, we get a major event that led to the origin of complex cells. And this ancestor here was really successful. All the living things on Earth that are more complex than bacteria can trace their ancestry back to that first endosymbiosis event. Now, it should be noted then, what endosymbiosis means is that we have to revise our tree notion of the history of life. So endosymbiosis forces us to revise the concept of the evolutionary tree. Now, trees have branches that diverge from the trunk, right? So if we start at the trunk, all the branches, as you go up the trunk, the branches separate, they diverge. But endosymbiosis means that cells or species converge to form a new kind of organism. So this picture of all the branches diverging is not quite accurate. Endosymbiosis theory requires that we have a diagram like this, where sometimes the branches on the tree of life can run into each other, forming a new type of organism. And it happens several times in the history of life, this first one being extremely important. So endosymbiosis means that the tree of life actually has points of convergence, two species uniting to form a new kind of organism. And finally, we'll end with one last question. Are mitochondria and chloroplasts alive? After all, if they both had ancestors that were free-living bacteria, shouldn't we think of them as being alive within the larger cell? Now, you recall that in our cell unit, we made the claim that the whole cell is alive, but none of the parts of the cell are alive. So here, do we have an exception to this? Is the mitochondria alive? Are these chloroplasts alive? And the answer biologists would give to this is no. And the reason can be seen in this diagram. Now here we have the nucleus of the plant cell. The plant cell has mitochondria and it has chloroplasts. Now scientists think in the case of mitochondria, the original bacteria that got incorporated, the ancestor of mitochondria, would have had maybe something like 1,000 genes. Genes are regions of DNA that code for proteins. But today's mitochondria, while there is a circular piece of DNA, there's only 13 genes. Many of the genes got transferred to the nucleus. And likewise with the chloroplast. So the cyanobacteria that may have been engulfed but not digested might have had 2,000 to 12,000 genes, but today's chloroplasts have 60 to 100 genes in the DNA. Again, many of the genes got transferred to the nucleus. So here we have a situation then that while originally they would have been, in a sense, living cells that just found a new home, over time, due to this transfer of genetic information from the mitochondria and the chloroplasts, they then become dependent on the host cell. So if we were to pluck out a chloroplast or pluck out a mitochondria, it would not be able to survive on its own. And for that reason, then, we're going to say no. The mitochondria and chloroplasts are not technically alive because they would not be able to survive and be self-maintaining outside of the host cell in which they reside.